Hey guys, so it's time for another video. So here we go. You'll notice this time I'm actually in the lab and not driving and being legal. So that's awesome. And this is where I work, right here. This is my place that I sit and I do my jazz. This is my computer. It's playing some music, and that's basically it. Right now I've just been doing a lot of reading, so that's pretty great. We've been reading up on axons and cephalopods and nervous system and sodium channels and all that great stuff. But yesterday we got to do some pretty exciting stuff. Wheelie chair over here. But we did, you can't really tell, looks really boring now, but yesterday we had 3 frozen octopuses and Becky Williams right here. Becky Williams is a friend and colleague of Shanna and Charles, and I guess me now. Yeah. And she actually studies blue ring octopuses. I don't want to say exclusively, but she studies them a lot. She actually has a poster over there that you can't see, but now you know there's a poster over there of work that she's done with blue ring octopuses and the TTX that they have. So she came in yesterday. She had some frozen octopuses, and we're practicing on them because we have the ones we ordered from Indonesia coming in hopefully next Tuesday. Who knows, but they're supposed to be here next Tuesday for our actual experiment. So to practice, she brought some in. She brought 2 in, and then she brought another species in. I don't remember what it was, but it was bigger, and it was kind of purple, and it was from Florida. So there you go. And we dissected them just to find out what it was like, because we are looking for stellate ganglia and optic lobes and the hearts in these octopuses. And the stellate ganglia, I guess I will explain to you what they are. Ganglion are cell bodies, so stellate ganglion are nervous cell bodies. And since we're studying TTX, which is this neurotoxin, it makes sense to take out stellate ganglion. And they kind of function as the central nervous system of an octopus. For us, our central nervous system is the brain and our vertebrae, the spinal cord. Obviously, octopuses don't have a spinal cord, so they actually have stellate ganglia. They kind of do the same jazz, basically. And so that's what we're studying. And then we're studying the optic lobe, which lies right behind the eye. I don't know if people have one, but they lie right behind the eye, and they process visual information, which is super important because octopuses have very, very good eyesight. They change color and all that, but they don't have cones. So we have cones, and that allows us to see all of the colors that we see. And then we have rods. Octopuses only have rods, so they can't actually differentiate between colors, but they can do that between contrasts, which is like crazy good eyesight if you can change your color based on the contrast of things, especially in the ocean. So we're studying that, and then we got out their hearts, because octopuses have 3 hearts. They have 2 brachial hearts, which bring in the oxygen from the gills, and then 1 systemic heart, which is actually doing the heart jazz. We couldn't find the systemic heart, but we did find the brachial hearts, so we took those out. And we're also doing some, because TTX is really high circulating in the blood, and then we took out some skeletal muscle just to test it. And so that's what we got. I didn't get to do the blue ring octopuses because this was my first ever dissection ever, and they're tiny, like they fit like in the palm of my hand, tiny. So I didn't get to do those, but I did get to do the bigger one, and I took out stellate ganglion, which you can see with your eye. Like, that's pretty cool. Cephalopods have really, really big nervous systems. So I used the dissecting microscope, but you don't have to. If I had known what I was doing, I could have done. And we took out the hearts, and then that was pretty cool. We did some work on them. Charles was running some RNA experiments. They didn't work out very well because these octopuses had been in negative 80 degrees Celsius for a while, so that didn't work out very well, but it was a dry run just to get everything done, practiced, and we know what we're doing now when we get the octopuses in a little while, to actually run some RNA experiments, which is super cool. Today, Charles is coming in sometime, and we are going to do actually some RNA. I'm actually going to do some RNA stuff. We're going to do some PCR. I don't exactly remember what that is, but I know that it's RNA and DNA, and there's gel, and you put it in some ultraviolet stuff, and it tells you things. And so that's what we're doing today, and that's pretty cool. I have pictures of our dissection, which will be on the blog. So if you want more information, a whole blog post, more information, and some pictures, check that out on Tumblr. I have the link on here. And yeah, that's basically it. That is my video for today. And it was great pretending to actually talk to a real person. Bye.